my name is Nick Weise, and in the last section we looked at the way in which candidate enzymes might be chosen for use and further application. In this submodule, we'll be looking at some of the ways in which such an enzyme can be studied in the laboratory. So it is often difficult to study an enzyme in its natural host organism, as it is unlikely to be present in reasonable amounts, and specific enzymes can be difficult to isolate for study. The organism itself may also be unavailable or difficult to grow under laboratory conditions. As such, we often use laboratory strain bacteria instead. Using molecular biology techniques, it's possible to introduce a gene encoding for an enzyme into an easy-to-handle bacterium using simple DNA manipulation strategies. A simple way of doing this is by constructing a circular piece of DNA, called a plasmid, and using this to genetically modify E. coli. By growing the genetically modified bacteria, it's possible to control the production of the enzyme in question and make useful quantities for further investigation. An additional feature, such as a protein tag or a leader sequence, can also be added to aid isolation of the enzyme. This will allow it to be studied independently of the cellular machinery upon isolation. Enzymes can often be studied without any further processing, however, if the cells in which they are produced harbour the enzyme. They can be studied in that way. In many biocatalytic applications, whole cells are added directly to a reaction mixture, whereupon the reaction occurs inside the cells, with the reactants and products passing freely across the cell membrane. However, in some cases, this cell membrane acts as a barrier between the added chemicals and the biocatalyst. In these cases, the cells can be broken open or lysed to create a cell lysate. In cases where an enzyme needs to be studied independently or a reaction is interfered with by other cell components, the enzyme can be isolated from the rest of the cell lysate using various biochemical separation methods. Some of these separate proteins based on their size, and others use affinity tags, which can be fused onto the enzyme when the gene is initially introduced. When an enzyme has been selected, produced, and is available in a suitable formulation, i.e. as whole cells, cell lysate, or in its isolated form, the activity of the enzyme can be assayed. This is often done to find out which chemical reaction the enzyme performs, and how well it does this under which conditions. It can be done via direct observation of the product formed, or by an indirect detection method. Examples of indirect detection include further processing of the product formed, or an enzyme cofactor that might be used to produce a measurable output, a color change or a pH change. An example of a pattern that might be observed with a color change, enzyme detection assay, with increasing activity, can be seen on the right. In cases where a simple assay is not available, the reactants and products may need to be separated and detected individually to see if the reaction has worked, and also to what extent. Examples include chromatography, whereby reaction mixtures are run through a column which separates compounds based on their chemical properties. This is commonly done in a flow of liquid, HPLC, or high performance liquid chromatography, or a flow of gas, GC, gas chromatography. After sample preparation, analytical techniques can be used to determine what each separated compound is, whether they are the reactant, the product, or possibly a byproduct of the reaction. These analytical techniques include NMR, nuclear magnetic resonance, or MS, mass spectrometry, among others. One biochemical observation of an enzyme's activity is the measurement of the kinetics of the reaction catalyzed. This is done typically using a set amount of the enzyme and varying the amount of substrate used. The rate of the reaction for each concentration of substrate is then measured, directly or indirectly, and the two variables are plotted on a graph. The data for most enzymes, which are said to display Michaelis-Menten kinetics, typically follows a curve. From the data pattern, two values, Vmax and Km, can be ascertained. Vmax is the maximum possible rate of reaction for a given enzyme with a given substrate. Km is the substrate concentration needed to achieve a rate half that of the maximum and is used as a measure of the affinity of an enzyme for its substrate. These two parameters will be different for different enzymes with the same substrate, and for the same enzyme with different substrates. As such, they are useful measures by which different enzymes can be compared. To allow the rationalization of enzymatic activity, the three-dimensional structure of an enzyme can be solved. The sequence of amino acids that makes up a protein and causes it to fold in on itself makes the structure of a protein very particular to the sequence involved. This fold pattern, shown on the right for a specific protein, places certain amino acids in particular orientations in 3D space, and thus allows them to fulfill certain functions. In the case of enzymes, the relative positions of the different parts of the protein allow it to bind specific chemicals and catalyze reactions between them. This means that if the structure of an enzyme can be visualized, it is often easier to infer how the enzyme works and what could be done to manipulate the reaction, by changing the conditions in which catalysis takes place, or even by altering the protein itself. One common technique for performing such structure analyses is X-ray crystallography. This involves the crystallization of a pure protein sample into a regular pattern and diffracting X-rays through it to ascertain the average structure of all the proteins in the crystal. Other techniques, such as nuclear magnetic resonance and electron paramagnetic resonance, work via the application of a magnetic field to a protein sample and analyzing the way this field is distorted by the sample. This is the same principle that is applied to body scanning equipment in the field of medicine, but on a much smaller scale. Samples of protein can also be frozen and analyzed under very powerful cryo-electron microscopes, which detect electrons reflecting from the sample rather than light, unlike the way optical microscopes work. In summary, in this module we've seen how molecular biology enables production and isolation of enzymes. Upon isolation of enzymes, enzyme activity can be studied either directly or indirectly using different assays. Also, measuring the kinetics of catalysis allow comparison between enzymes or comparisons of the same enzyme under different conditions or with different substrates. Finally, we've learnt about how structural biology allows or can allow the rationalization of enzymatic activity by looking at the three-dimensional orientation of the different amino acids in an enzyme.